Good morning. Today's lesson, I guess, or discussion is about the structure and the chemical makeup of RNA. We're going to look at how RNA differs from DNA, okay? So remember, just a gene is just a section of DNA that codes for a protein, okay? That's really the whole goal of DNA, is to make proteins, and it has to go through using RNA to do that. So chromosomes contain thousands and thousands of genes, and we build a huge assortment of proteins off of that, okay? So that's all DNA really codes for. And DNA, like I said, is a code for that protein. We're going to get into like RNA and protein synthesis in this week. We need to understand like what the central dogma is considered, and that's really DNA going through transcription and translation to make a protein, okay? It's just called the central dogma because a scientist called it the central dogma one time. But all it is how DNA codes and goes through transcription to mRNA, and then from mRNA goes through translation to make a protein. All right, let's get into this real quick, though, okay? So DNA has thymine, adenine, guanine, and cytosine, all right? And it's double-stranded. RNA has uracil, all right? Cytosine, guanine, and adenine. Your RNA does not have thymine, all right? And you can sort of see double-stranded DNA, single-stranded RNA. All right, so we're going to get into this. So the function of RNA is to make proteins, right? And it's made up of the same things. The nucleotides are the same things. It's a phosphate and nitrogen base, but it comes with the sugar is ribose, not deoxyribose. A big thing here, two things really, is it's single-stranded, not double-stranded. And instead of thymine, RNA has uracil. And also remember, DNA is located in the nucleus. RNA is located in the cytoplasm, right? So we're going to get into that. I would watch this YouTube video here if I were you guys. It's a good explanation. There's three types of RNA. There's mRNA, also known as messenger RNA. This RNA goes into the nucleus and codes the DNA and then comes back to the ribosome, okay? Then there's rRNA. It's also known as ribosomal RNA. This is RNA that makes up the ribosome. If you guys remember from earlier in this semester, the ribosome is where proteins are made at. And lastly, there's tRNA. That's transfer RNA. This RNA brings down amino acids to pair up to make a strand of amino acids, which makes a protein. All right, we're going to get into that tomorrow. So here's mRNA. You have rRNA making up that, and then tRNA bringing down the amino acids. All right. So a little comparison chart. DNA is a T, A, C, G. Okay, that's the codes of DNA. The complementary strand of DNA would be to the A would be T, to the T would be A, C, G, and GC. mRNA is very much like the complementary strand to DNA, all right? Except the T's are U's, all right? So if the DNA is A, an mRNA is going to code it as a U, a T, A, C, G, and GC. So the only thing difference here is this T and U here, all right? And then tRNA actually codes itself off of mRNA. I know we're going to get into this a lot tomorrow and Wednesday. But tRNA is almost identical to DNA, whatever the DNA strand looks like, except for the U's and the T's. See, T turns into a U. We're going to get into this a lot more when it comes to tomorrow and Wednesday, when it comes to protein synthesis. But this is a really helpful chart to get through all that. But today, the big thing is just knowing the structure of RNA. Right? Big things, single-stranded, and it has uracil instead of thymine, and it's located in the cytoplasm. All right, and knowing the three types. There's mRNA, rRNA, and tRNA. We will get into protein synthesis and the processes of transcription and translation a lot more tomorrow, okay? So we're going to have a Zoom meeting tomorrow, not today. All right, I'll talk to you guys later.